but under different regulatory conditions at the time. And since then, products have become very complex and are evolving very, very quickly. I won't go through all of the different types. There's so many different types. A few of them are given on this slide. Particularly later, we'll talk about chimeric antigen receptive engineered T lymphocytes, just as an example. Essentially, all of these different medicinal products that are coming through clinical trials at the moment involve complex scientific platforms, and rarely the mode of action is elucidated for these products, which can become a problem for regulatory matters. So an ATMP, essentially from a regulatory perspective, an ATMP can be divided into three different categories. And these are known as gene therapy medicinal products, somatic-cell therapy medicinal products, and tissue-engineered products. And these are essentially categorized on their mode of action and how they are prepared. With GTPs, for example, they are normally naked DNA or encapsulated DNA that to deliver a gene that might be missing, and they're involved in regulating, repairing, replacing, adding, or deleting a genetic sequence, particularly in medicinal context. Somatic-cell therapies, they are subject to substantial manipulation of the tissue in the manufacturing process and have a view to treating or preventing through pharmacological, immunological, or metabolic action using tissues or cells. And tissue-engineered products, they're usually a mixture of cells or tissues that are organized, and their intention is to replace an organ or tissue in the body. So in Europe, there is a very well-defined regulatory strategy pathway for regulation of these products. And these are regulated under the centralized procedure by the Committee for Advanced Therapies and the CHMP under EMA. And they're governed directly by two key directives and one key regulation, 2001-83 EC and 2009-120 EC as updated, and 34-2007 EC. Now, I won't go into further detail on those, but essentially when you're looking to regulate these products, they are your key directives that you should begin with. The other factors that come into play are Directive 2004-23, which relates to the donation, procurement, and testing, i.e. the safety of the source materials for these products. So I won't say any more about that slide, but the slides will be available, I understand. So the key committee for regulation or assessment of these products within Europe is the Committee for Advanced Therapies, which is otherwise known as CAT. It's responsible for assessing the quality, safety, and efficacy of ATMPs and for following scientific developments in the field. Now, the committee is made up of CHMP members, patients and clinicians and experts in the field. So it's a very broad committee that has a very broad interest in this area and keeps up to date with current knowledge. The key interactions that you can have for these types of products in Europe, and they're very recommended that you speak with the agencies and the EMA during development of these products, are essentially SME or small medium enterprise discussions to understand whether the company will benefit from financial benefits during development, task force meetings, scientific advice, and classification and application of the product. And essentially, as I mentioned, small medium enterprise meetings or classification in effect can help with reduction of fees. In addition, task force meetings are essentially to understand or to get feedback from the agency, particularly, on the scientific design and the development pathway for your product at very early stages, quite often non-clinical. Product classification is very important for these products insofar as it's necessary to understand where a product falls within the regulations so that you can find the correct pathway to be able to understand the data that's necessary to complete clinical trial applications sufficiently and later marketing authorization applications. So it's very recommended to request a product classification with the CAT. And scientific advice would allow essential feedback on the development plan during the development of any of these products. I recommend to attend EMA workshops and interested party meetings so that you can educate the agency on the type of product that you might be developing. One of the new approaches that the agency, the EMA particularly, are encouraging at the moment, and it's written into the regulation that governs these products, is to use a risk-based approach. And certainly that can be used as an efficient tool to optimize your product development. Insofar as if you as a sponsor understand the risks that are associated with manufacture and clinical implications of your product, then you could go back and narrow down, reduce the amount of work that you need to do to provide useful data of your development plan. And that risk-based approach can then be discussed with the agency and through a scientific advice meeting, for example, to get buy-in from them during the development procedure. So that when you come to your phase 3 clinical trials or marketing authorization application, for example, the agency are familiar with your product and have agreed with your reduced development planning. There is still a lack, because of the complexity of these products and because of the lack of authorization knowledge of these products coming through the agency, for example, there is the tendency for the agencies to from the side of being conservative and to ask for a lot of studies, whereas they might not potentially be necessary. So it falls on the sponsor to use a risk-based approach. I'll move on to other aspects of safety, and a key one obviously is your quality dossier. And of course, ATMPs in themselves are extremely heterogeneous and complex in nature. And agencies that are assessed on a case-by-case basis because of this. Now, one of the ways to ensure their safety, of course, but also to assure that the agency understands the safety of the product and how the product will behave in the body, is to control the CMC, the quality part of the manufacture of the product. And there are some key areas of that. Certainly characterization, and these key areas, if I can add, are particularly difficult to put together to understand for these types of products, for cell-based products. Particular characterization, it's very difficult. You have a mixed cell population to understand what the identity or the purity of your product is. Viability is usually quite an easy one, but certainly which cells are responsible for the mechanism of action in the body is often quite difficult to elucidate. Likewise, with the impurities, if you have a group of cells that are responsible for the mechanism of action, then the cells that are not responsible become impurities. And sometimes what cannot be justified, there is some cause for the agency to request that cells are removed from the final product, which obviously can be quite a difficulty. It's important to understand what are the active substrate and what part of the product are impurities. Biological safety speaks for itself, as does viral safety. these tissues are coming from humans. They are potentially being mixed with fetal calf serum and other animal-derived substances. So viral safety is key. And its robustness, you need to understand that each batch that you make is consistent. So when the batches are tested in humans, in clinical trials, for example, and later in commercial situation, that the product itself is reproducible and replicative, so that the medicinal effect can be deduced back to the product in an effective way. And just one point, that GMP for these products is slightly different from normal medicinal products, and it's aligned in Annex 2 of the Eurodirect guidelines. So I'm going to speed up because I'm taking up time. So just to focus in on those, the identity of the product, as I mentioned, is quite important, and certainly it's necessary to perform a phenotypic analysis of the product according to surface markers of the cells, for example. I'm going to move through these quickly. Again, you will have these available. Purity, I've already mentioned. There could be a reduction, elimination of undesired cells in the product. But there are also factors relating to the process that could be classed as impurities. And certainly, taking products from laboratory, say in a university, through to a startup company, for example, where there may already be a proof of concept, understanding the reason for adding a number of growth hormones, for example, is necessary, because at the end of the day, if those growth hormones are not doing anything practical in the process, then they could be seen as an impurity, especially if they still remain in the final product. The potency assay is critical, and it's related directly to the mechanism of action of the product, which in cell-based products, for example, can be difficult to elucidate, as we previously mentioned. So the potency assay can be quite difficult to define, particularly in early stages. However, it's very important to have in place in phase 2, at least before phase 3, insofar as it's necessary to be able to understand the what we call dose of the product, how much of the active substance is going into the patient, and also for comparability of batch to batch to understand that the activity of the product is comparable. Viral safety, again, viral contamination can come from starting materials or raw materials. So it needs to be controlled through the manufacturing process, and there are guidelines describing how to do this. Particularly, one thing that's interesting to note is that viral safety requirements can be different in Europe versus the US. And it's necessary to check when you're bringing a product from the US to Europe, for example, that the necessary donor testing and viral safety studies have been performed. Now, slide. Just on quickly to the non-clinical development. There are a number of specific challenges related to cell-based products and the non-clinical area, and particularly selection and availability of appropriate animal models. Obviously, where you have species differences, cell-based products can behave very differently in a different species, and therefore finding a model that represents the human scenario is quite difficult. Likewise, with biodistribution, cells don't necessarily home to the same organs in animal, and also following those cells can be quite difficult. So those are issues that need to be addressed. Persistence of the cells needs to be identified, measured, and tumorigenicity is obviously an area of big concern, particularly with stem cell products. So a minimum plan for non-clinical would be proof of concept, biodistribution, cell persistence and differentiation, and tumorigenicity. Now, depending on the risk-based approach again, toxicity studies may be needed. There is some information on the selection of the animal model. Essentially, it's useful to animal model that you can assess efficacy and toxicity, if necessary, in the same model. And obviously with the route of administration as you would use in the patient. I will move on. I shall move on from that one. So tumorigenicity is a key safety aspect, as I've mentioned. And you may find that you might be asked to produce both in vitro and in vivo data, particularly for karyotype analyses of the cells, expression of surface antigens, telomerase activity, and population doubling. And the histology becomes a very useful tool. Related to clinical, just very quickly, key challenges there with cell-based products are related to the targeted indication, the population, and administration, which, to be honest, is similar between most medicinal products. With these products, the issues become with the long-term safety and follow-up, depending on the persistence of the cells, and how long do the cells last in the body, and what do they become, and what do they influence. So that needs to be assessed. Possibly of randomized double-blinded comparative studies, that becomes more difficult with these products as well. So just some take-home messages. This field is evolving very, very rapidly, and each case is different. The agency knows that, and obviously one size does not fit all, so it's necessary that the products are well characterized and understood. Regulatory expectations are continuing to adapt. That's the case. CMC non-clinical development data must be tailored to the nature of the ATMP and adopt a risk-based approach to determine what studies and testing is necessary. And interaction with the regulatory authorities via scientific advice does provide support for suitability of the development plan to obtain a marketing authorization once complete. Now, I haven't mentioned any guidelines through this presentation. This is something that we can provide to you if you would like. There's a wide range of guidelines covering all of the different types of products, and they do provide excellent guidance on the type of data that's necessary for authorization of these products. Okay, thank you.
For this summary of European Regulations and Directives and the transition to non-clinical development. Now let's see how to optimize product development at the preclinical stage. François, please go ahead.
Thanks, Maud. So my talk will be regarding preclinical evaluation of cell therapies. I will shortly introduce the situation in oncology, and after that I will describe what kind of study are needed to perform a preclinical evaluation of this type of therapy, and I will describe some case using CD19 as target. So first, a short introduction. Cell therapy are from long time ago used for non-oncology fields, so mainly for regenerative medicine, using mesenchymal stem cells transplantation, stromal cells, or even hematopoietic stem cells transplantation. And in this oncology field, these cell therapy are used for many diseases like heart disease, immunology, or traumatic brain injury. In oncology field, there are mainly two cases of therapies. One is whole cell vaccines, cells from very various origins. That could be from yeast, viruses, or human cells, and we'll focus on adoptive T-cell therapies, which is really growing case in oncology. So what is adoptive T-cell therapies? It's a modification of the T-cells to generate cytotoxic activity in an HT-independent manner. So the objective is to implement a new target on the T-cell receptor, so a fusion between an antibody and the TCR subunit. So this is known and historically referred to as T-bodies. As some proof of principle with T-bodies, these T-cells were improved, and the structure, mainly the intracellular part of the receptor, has evolved with various generations of chimeric antigen receptor. So first generation used an CFV fragment of antibody linked to an intracellular domain and then fused to a signaling motif of the T-cell iota receptor chain. Second and third generation of CAR T-cells are using an stimulatory activating motif in order to improve the expansion, the proliferation, and the efficacy of T-cells. So the second generation is for have only one costimulatory activating motif, and the third generation shows the incorporation of two costimulatory motifs. This in the objective to increase the proliferation, the cytotoxicity, and the persistence in vivo of the transferred T cells. There is some prerequisites in order to select the best targets and to select the best pathology to test these cell therapies. Regarding the tests, there are clear need for having a really high affinity antigen binding domain and a really restricted expression on the tumor cells and quite no expression on normal tissue in order to avoid off-target, off-tumor but on-target effects that be linked to critical side effects. Regarding pathologies, there is now really nice demonstration in clinic for hematological malignancies, and targets in this case are CD19, 20, 22, or 30. For solid tumor, there is also active development in this case using targets like GD2, R2, EG4 receptor, IL-13, or even PSMA. For the preclinical evaluation, there is specificity regarding the experimental system, where we need an intact immune system, also a tumor target. So this is related to tumor microenvironment, and also we need clearly to establish assays for evaluation of the immune response, so the quality of the transfer system. To be able to perform such a kind of experiment, the husbandry, so the animal facility, needs to be biosafety level 2, and authorization to use human samples are also required. So, in short, all the assays are dedicated to characterize the transfer system, so the cells, the T cells, and so type of the T cells, and also the functionality. So the T cells are able to produce cytotoxicity and specific cytotoxicity against tumor. Does the T cells release cytokine, and what kind of cytokine? And is these T cells able to proliferate in vivo or in vitro? So these three parameters are can be tested in vitro using classical assays, so chromium release assays, cytokine release assays, or proliferation assays. In all these vitro assays, non-transduced T cells, so negative control, are if of interest and really useful, and also negative tumor for the antigen, target antigen is also useful to test the specificity of the CAR T cells. So in the in vivo situation, there is various assays, various models that could be used. So completely syngeneic models. However, in this case, there is a need for a surrogate system. So it's mouse tumor targets, it's mouse immune cells. However, there is mainly advantages regarding the complexity of this model. There is a possibility to analyze the off-tumor on-target effects. There is also a possibility to evaluate the conditioning of the animals before the transfer of these T cell therapies, and there is also a possibility to analyze the immune interaction with the immune component of the mouse. The second possibility is to use a chimeric system using immunocompetent mice modified to express the human targets. So in that case, there is some drawbacks, mainly the capacity of the mouse to mount an endogen response against human antigen, so a foreign antigen as compared to the mouse system. And also there is possibility of autoimmunity response or peripheral tolerance induced in this case. This model use xenogenic models where this transfer of human transduced T cells, or the CAR T cells, in tumor-bearing, so human tumor-bearing immunodeprived mice. And in this case, the drawbacks are mainly the capacity to have graft-versus disease from the transferred T cells, and also lack of endogen lymphocytes that could affect the response to T cell therapies. So we'll take the example of the CD19 CARs in order to highlight all these three different experimental systems in vivo. So I've selected few papers from the literature, and the first one is to illustrate a fully Syngeneic model. So this is the results from a paper published in Blood in 2010, and the model used in this case is a brain lymphoma. The tested therapies is the second generation CAR. So using an ATMP for the murine CD19, a co-stimulatory activating motif of the CD28, and for sure the cytoplasmic portion of murine CD3 iota chain. And in this case, the effector T cells are then mouse T cells purified from splenocytes and then transduced with this second generation CAR. The main finding in this case is that the CD19 CAR T cells are really effective to target lymphoma cells, but also the normal B cells that for sure express CD19. This model was tested ISC, and effect of transduced T cells are seen in both cases, so there is no problem for biodistribution in whole the body of these transduced T cells. And the author has tested the conditioning of the mice in order to evaluate efficacy of the CAR T cells in whole body irradiated animals or not. And there is really a critical factor to the efficacy of CAR T cells when mice were whole body irradiated. They also have compared the efficacy of monoclonal antibody against CD19 with CAR T cells, and they have demonstrated that CAR T cells are really superior to the monoclonal antibody. So the second case is a fully xenogenic model, so immunodeprived mice injected with human lymphoma. And in this case, author has again used a second generation CAR T cells, where the scAV are coding for CD19. Again, the co-stimulatory activating motif is from CD28, and the cytoplasmic region of the human CD3 iota chain. In that case, the T cells are coming from healthy human PBMC donor and transduced to express this CD19 CAR specific for CD19. So in these results are really relatively limited. So the only critical finding is that they show an effect of the T cells. So the therapy is really effective, and they are also able to demonstrate a link between the persistence of these T cells in vivo and the consequent, so the efficacy of these transduced T cells against this lymphoma. There is no demonstration of relationship between the number of T cells and interaction with the mouse immune systems. There is no possibility of tumor on target effect in that case. So xenogenic models are somewhere limited as compared to the mouse Syngeneic system. The third case is an illustration of the chimeric Syngeneic model, so meaning immunocompetent mice expressing human targets and transduced cells modified to express the scAV specific for the human antigen. So in this case, the model is the E4 mouse thymoma that were tried to express human CD19. The host is transgenic black cyst mice that express human CD19, and the tested therapies is first generation CAR T cells composed where scAV are against CD19, human CD19, and the cytoplasmic region is from mirroring CD3 iota chain. In that case, again, the effector cells are from the mouse, so the T cells from the splenocyte. So again, authors have demonstrated that conditioning of the animals before transfusion of CAR T cells really affect the efficacy of these CAR T cells. So they have tested cyclophosphamide for preconditioning regimen. They also evaluate the composition of these T cells, meaning the number of CD4 and CD8, and they have demonstrated that both types of T cells, transduced CAR T cells, are required for optimal efficacy in vivo. They also have demonstrated that the efficacy in vivo is fully dependent on the IL-12 stimulation. So, as for conclusion, so there is maybe 3 cases in vivo, and type of model have limitations and drawbacks. So it's really difficult to select one to perform all the preclinical evaluation. However, knowing that there is many clinical trials that test T cell therapies in hematological malignancies, but also in solid tumor, these evo systems need to be improved or to be understood in depth in order to help to the better definition of the product that need to be tested in human. So I'd like to thank you for your attention.
Okay, François, Amanda, thank you very much for this overview of regulatory strategy and preclinical technology to evaluate cell therapies in oncology. I hope that the audience that we had today appreciated this presentation. So, Hamda, there were a couple of questions that came up to your presentation, and I just want to use the next 5 to 10 minutes to address some of these. So the question is, in your experience, what are the key regulatory barriers or point of objection that companies face when to take their ATMP from the lab into the clinic?
Okay. One of the ones is source material. It's quite difficult when you're producing or manufacturing a cell-based product to find sufficient or adequate source material. So that can be difficult, particularly in scale up of the product towards your phase 3 studies. Secondly, as I mentioned earlier, characterization of the product can be one of the key aspects that can be difficult, insofar as understanding the mechanism of action and therefore understanding what moieties in your final product are actually active, are actually responsible for causing the medicinal effect. And of course, then developing the potency assay on the back of that can be quite difficult. And likewise, comparability between the batches. I mean, with cell-based products, for example, particularly stem cell-based products, the products are differentiating and proliferating during your manufacturing process and during the time that they're in stability, during the shelf life, for example. So how do you characterize those products? At what time point do you say this is the final product and this is the drug substance, for example? So specific challenges, which, when combined with the science and the regulations, can be quite complex and quite difficult to deduce. Personally, I think the fact that the cell products are changing means you have to fix a very, very short shelf life a lot of the time, which means that delivery of that product, released product to the patient, can be quite challenging in itself. So there's a number of different issues that are challenges from the lab, particularly to a regulatory scenario.
Okay, thank you for this answer. Then another question that I would like to raise, do the incentives from EMA for SME also count for non-profit organizations supporting development of autologous cellular immune therapies?
It's a good question. Yes, I believe so. There are certain requirements to fulfill the SME categorization, and one of those is that the turnover of the company is below a certain mark. So a non-profit organization, you'd normally expect to fit into that category, and it depends on the size, the number of people within the company as well. So if it was a small non-profit company, then yes, it should be able to benefit, is my understanding.
Okay, thank you. Jean-François, I have a question for you too. What is the appropriate lab environment to perform a preclinical study to evaluate cell therapies?
So there is first that should be hot to use human 5 cells. So there is authorization for GMO use, and means that it is T, so it's active T cells, it's modified T cells, so the lab should be also BSL2 authorized. So one requirement for the use, but also one for the in vivo evaluation, but another requirement for the manipulation of these man-derived cells.
Okay, thank you. Amanda, I've got another question for you. Can you discuss the difference between the ITF meeting and the scientific advice meeting, and what are the advantages and disadvantages of each?
Yeah, okay. So the ITF meeting, Innovation Task Force meeting, it's a very informal meeting. It's a really nice way for a company to introduce their product and the company to the EMA. And it's usually held around phase 1, phase 2s, not much later than that, because this specific meeting is not usually, well, they don't like to discuss clinical issues. It's really to discuss scientific and non-clinical issues. And it's a good way for the agency to get to know your product and what the potential, to discuss the potential risks at a very early time point. So it's a great basis to start the development plan. Scientific advice, on the other hand, is a much, much more formal process. There are a series of timelines that have to be adhered to. A letter of request has to be written that is in a format that is on the EMA website. You go into a scientific advice meeting with a specific set of questions that are worded in a specific way, insofar as you will gain a specific answer. So the whole process is extremely formal. You have to build a briefing document to support all the information, so there's a certain amount of work involved. And at the end of the day, the answers, although officially non-binding, in fact, they are reasonably binding, insofar as if you deviate from scientific advice, the agency would want to know why you didn't follow their advice, so you'd have to just make a justification later. And the other thing is that with a scientific advice meeting, there is a cost involved, of course, and if you wanted to ask clinical and non-clinical questions, particularly clinical questions, then the price is around 80,000 euros. So obviously it's worth to look into getting financial benefits that come with SME or orphan drug positioning, for example. But scientific advice definitely gives you, so a benefit is that the agency will give you specific information of what they want to see in, say, a marketing authorization application, and then both parties should adhere to that advice. So to be able to help your application when you get there.
Oh, thank you. The last question that I have, and I would like to apologize to anybody whose questions could not be answered. There were quite a few. We will get back to you with details later. So the last question I just want to pick up is, and it's quite broad, which studies are expected to be performed, need to be ATMP compliant, and which do not?
Subject to?
Which are expected to be performed with GE and which do not?
Okay, is that for me?
I think it's for you.
I can take it, that's okay. So I'm assuming we're talking about during the manufacturing process or development. Is there any more context to the question quickly?
No, I don't have more context. Right?
Okay, no problem. Okay, so DLP studies. Okay, there's two areas that these, well, particularly three areas that these are important. Certainly when you're generating proof of concept information in very early development stages, before you get to your fixed manufacturing process, GMP manufacturing process, anything you do before that want to document in a traceable manner should be done to GLP. Any laboratory work that you wish to be traceable in the future that could be used as useful data, that should be done to GLP. Non-clinical studies, your animal studies, again, should be done to GLP. That's very important because that data needs to be audited at a later date. And any analytical methods that are used for characterization, yes, but for during the manufacturing process, analytical methods are used either for release or for impulse controls, things like that. They are part of eventually become part of the GMP process, but they should be performed to GLP and certainly designed to GLP.
Thank you very much, Amanda. I want to thank everybody for attending this webinar today, and I wish everybody a good day. Bye-bye.
Thank you.